Have you ever thought about what truly makes you, you, right down to the molecular level? Well, the answer lies in molecules called nucleotides. So, they are microscopic units that hold the code of life. They are the building blocks of DNA and RNA. And today, we'll explore what they're made of and how they come together to form the molecules that define every living thing. So previously, we talked about nucleic acids and their importance in genetic information. But today, in this video, we will be talking about the components of a nucleotide. So, by the end of this lesson, you will be able to define nucleotides, describe how these nucleotides form, and demonstrate its naming convention. So let's get to it. Nucleotides are the monomers of nucleic acids, much like how monosaccharides form disaccharides. DNA and RNA are long chains made of these repeating nucleotide units. Nucleotides do more than just build structure. They carry genetic instructions, control protein synthesis, and participate in energy storage. Let's break down what makes a nucleotide functional. So the first component of a nucleotide is the phosphate group. The phosphate group in a nucleotide is derived from phosphoric acid, which is your H3PO4. But in nucleotides, it exists in its ionized form, usually carrying a negative charge, which helps nucleotides link together and gives DNA its acidic properties. So next is the pentose sugar. So from its name, pentose, it is a 5-carbon sugar. In your RNA, it is a beta-D ribose, but in your DNA, it is a beta-2' deoxyribose, which lacks an hydroxide group or an OH group at the 2' carbon of the sugar. So these small structural differences determine whether a nucleotide becomes part of your DNA or your RNA. So numbers with a prime, the apostrophe, 1', 2', refer to sugar atoms, while numbers without a prime or without the apostrophe, it only has a number. So this refers to the nitrogenous base. That detail matters when we talk about bonding. We're finally down to our last component of a nucleotide, which is your nitrogenous base. These guys serve as the letter of your genetic code. And there are two specific types, which are your purines and your pyrimidines. Your purines have a double ring structure, and it is composed of your adenine and your guanine, while your pyrimidines only have a single ring structure and are composed of your cytosine, thymine, and your uracil. Now, remember this. Your DNA only has your adenine, guanine, cytosine, and your thymine, while your RNA is the same but except your thymine. It is instead replaced by your uracil. So again, your DNA only has your thymine, and your RNA only has uracil. DNA for thymine, RNA for uracil. To form a single nucleotide, we will be needing the three components that I just discussed. First, let's go to your nitrogenous base. So if you remember, in your purines and pyrimidines, there are specific numbers which indicates the position of each element contained in the rings. So for your pyrimidines, for the first nitrogen, which is your N1, it links to your 1' carbon of the pentose sugar. And if it's purines, the 9th carbon, your N9, will link to the 1' carbon of the pentose sugar. And this linkage, it is a condensation reaction. So they come together and they make a bond called your beta-N-glycosidic bond. So this structure is now called a nucleoside. Not quite a nucleotide yet. So it's a nucleoside with an S. So in order to make a complete nucleotide, you will be needing one more component, which is your phosphate group. Now the phosphate group will be linked to the 5' carbon of the pentose sugar through a bond called a phosphoester bond. Again, a phosphoester bond. And now you have a complete nucleotide. But do remember that nucleotides may carry one or more phosphate groups. So if it's just carrying one phosphate group, it will be called monophosphate. If it's two, it will be diphosphate. And if it's three, it will be triphosphate. So do take note of these naming conventions because we will be using them on how to name a nucleotide. Now let's learn how to name your nucleotides. To name them, we first have to name the nucleoside. So remember that your nucleoside doesn't have a phosphate group yet, so it's not a nucleotide yet. So let's go to your purines, your adenine and your guanine. They end with an INE. That INE will be replaced with an OSIN. So adenine to adenosine and guanine to guanosine. Now for your pyrimidines. So for your cytosine and your thymine, they both end with INE. So let's change that to idine. So from cytosine to cytidine or cytidine, and your thymine to thymidine or thymidine. So for your uracil, your acyl will be replaced with the idine. So from uracil to uridine. If the sugar is a deoxyribose, you just simply add a prefix deoxy in front of the nitrogenous base. So example, your adenosine. It will now be deoxyadenosine. So it applies to all of the nitrogenous base except your uracil, because your uracil is for your RNA, and RNA only has a ribose sugar, not a deoxyribose. Finally, we just have to add the appropriate suffix, which is, like I said before, your monophosphate, diphosphate, and your triphosphate. It is a suffix because it is placed at the end of the name for the nucleotide. So you have an example here, your ATP. So your ATP is known as your adenosine triphosphate. So it has an adenosine, a nitrogenous base, a ribose sugar, so it doesn't start with deoxy, and it carries 3 phosphate groups. So it's a triphosphate. Hence, adenosine triphosphate, or your condensed name, which is your ATP. Here is a table of nitrogenous bases for your RNA and your DNA, and how differently they are named if they become a nucleoside and a nucleotide. So in your nucleoside, it ends with osin or idine, and eventually it will change if it becomes a nucleotide. And here we have the structure of those nucleotides. This is what they look like. Now we have ended the lesson. Woohoo! So we will now be playing a game that will test your knowledge whether you've learned something from this video or not. So let's play nucleotide this, nucleotide that, where you'll be guessing the component based on the provided image. If you need more time to answer the question, you can just pause the video. So we have your first question. Is this a phosphate group, sugar, or a base? If you guessed phosphate group, then you are correct. Now let's go to our next question. What sugars are shown in the image below? So the left one is a deoxyribose, and the right one is a ribose sugar. Now to our next question. What bond is indicated by the arrow below with a question? If you guessed beta and glycosidic bond, then you are correct. Now for our last question. Name this nucleotide. The name of this nucleotide is deoxythymidine 5' diphosphate. Let me know in the comments below how well you did on this activity. So if you did well, congratulations. And if you didn't, it's okay, you did your best. we can just go back to the video and relearn what we haven't mastered and go back to this activity and answer it again. So we have another activity where we apply what we've learned, and I will be asking you what's wrong with the naming of the nucleotide. So here is your structure. Now, what seems to be wrong with the name? If you guessed deoxy, then you are correct, because deoxy shouldn't be there in the name. Why is that? Because the sugar in the structure, or in the nucleotide rather, is a ribose sugar. It is not a deoxyribose. And if you remember our naming rules, if it's a deoxyribose, you add the prefix deoxy at the start of the name. So, instead, the name should be guanosine 5' monophosphate. We are almost finished with our video, so now let's have a recap of the lesson. And I will be asking you a couple of questions. See if you remember what you've learned. So, what are the 3 components of a nucleotide? So yes, these are your phosphate group, your pentose sugars, and your nitrogenous bases. Now for your next question, what is the difference between a deoxyribose and a ribose? So a ribose has an OH group on its 2' carbon, while a deoxyribose only has a hydrogen. Next question. What's the difference between your purines and your pyrimidines? So your purines have a double ring structure, that is your adenine and your guanine, while your pyrimidines only have a single ring structure, and these are your cytosine, thymine, and your uracil. Next question. What type of bonds connect nucleotide components? So these are your beta and glycosidic bond, which connects the base to the sugar, and your phosphoester bond, which connects the phosphate to the sugar. Now for your next question, and last one, how do you name a nucleotide? So you use the nucleoside name, add mono, di, or triphosphate, and add deoxy if it contains a deoxyribose. To test how much you've learned from this discussion, here is an assessment for you to try. So answer this on your own and check if your answers are correct at the end of this video. You can pause the video again if you need more time. And after you're done, you can submit your answers through this QR code, which will direct you to a Google form sheet. Thank you so much for joining us today. Stay tuned for the next topic, which is about pentose sugars. And subscribe for more videos about biochemistry from Genius Review Center, where we don't just study science, we break the code of science the genius way. Bye!